Good evening, everyone, and welcome to this special event. We are glad you could join us to celebrate surpassing $1 billion in annual research funding and to explore some bold visions looking forward. My name is Ana Lucia Cordova. I'm the Strategic Initiatives Manager at the Office of Research. I will be your MC for tonight, and we will start with some opening remarks from Prasant Mohapatra, our Vice Chancellor for Research.
Thank you, Ana Lucia. It is really a pleasure to see everyone here together to celebrate this momentous achievement. The goal of reaching $1 billion in annual research funding was aspired several years back. It is an accomplishment which has been, which can be claimed by only 20 universities, 20 public universities in the nation. So we are in that group of universities that belong to that highly performing category. While the number itself is just an index, in the simplest form, the real intent and the value surrounds our missions to deliver insights, solutions, and innovations that improve quality of life and promote a brighter future for all of us. Our accomplishment along that path shows up in other key indexes. For example, last year alone, we enabled the launch of 13 startup companies. We processed 132 records of inventions, 269 patent applications, submitted more than 4,000 grant proposals, and published more than 8,000 papers. That's just a glimpse of our research accomplishment during just the last year. And those are just a few of the related numbers, but most important is the global impact that emerged from our research. UC Davis has a rich history of foundational discovery and impact. We have changed agricultural practices, increasing food supply, efficiency, and sustainability. We have pioneered new medical treatments, diagnostics, and preventions. We have shifted the transportation model in ways that protect our health and the health of the planet. We helped enable huge advancement in digital communication, and we have unlocked insights and programs to enable social equality and well-being. We thank the entire research community for reaching beyond what is expected in order to achieve this impact. Everyone, each and every one of you played a critical role in our growth and the related impact, from idea to execution, from policy to compliance, from recruitment to closeout, from proposal to negotiations, from data collections to analysis, IP protection to commercialization. You all have played an important role in making all of those activities. To our faculty, researchers, staff, and students who lean into the challenges the world faces in order to provide answers and solutions, we thank you. To research community in the supporting role across the entire campus, you ensure that vision becomes a reality with pride and a commitment to excellence. We thank you. To my colleagues in the Office of Research, despite not growing in size at a proportional rate to the amount of funding, you continue to go above and beyond, providing exceptional support, skill, and knowledge that is truly world-class. A very personal and special thanks. You all have been an inspiration for me. Thank you very much. I would also like to acknowledge some of our previous Vice Chancellor of Research. I think at least two of them are here. Harris Lewin, Cam Carter, and Barry Klein, all of whom have held this position and have played a role in achieving this goal. I also want to give a shout out to Paul Dodd, former AVC. He flew all the way from Dublin, Ireland in order to attend this function. And to our Chancellor, Provost, Deans, and the leadership team, we thank you for all your support. Without your support, we wouldn't have made this feasible. As we look forward, we see the accomplishment of today as a stepping stone on our path to even better and bigger things. We will get a glimpse of some of those bold visions with many of the visionary research talks that have been planned for tonight. These will be very short talks. It is an exciting time to be at UC Davis, and I look forward to working with you all to continue our impact. So we are not, since this is all home crowd, we are not going through formal introductions. Now I would like to request Chancellor May for his remarks.
Thank you, Prasant. Good afternoon, everyone. I'm starting to really enjoy this particular room. Some of you may recall a couple of weeks ago, we announced our $50 million gift from the Resniks to the College of Agriculture and Environmental Sciences. So this is kind of the money room now. So every time I come in here, I expect to have some big money announcement. Anyway, great to be here for this particular celebration. Let me just begin by shining the spotlight where it belongs, which is on all of you. Together, you helped UC Davis achieve the greatest year for external research funding in UC Davis history. Surpassing $1 billion in research awards is no small feat. It is a significant milestone that, as Prasant said, fewer than 20 public universities in the nation can claim. I'll just add parenthetically that UC Berkeley is not one of those universities. Just saying. Somebody's clapping over there. I'm a Berkeley alum, so I can say that. Anyway, none of this would be possible without all of you. I'm grateful for your dedication and hard work that helped us to reach this level. In particular, I want to thank Prasant personally and everyone in the Office of Research for the crucial role that you played in supporting our research enterprise and advancing our mission. Thank you also to our exceptional staff who supported the professional staff in Office of Research. I want to acknowledge, as Prasant already has, the successful efforts of previous leaders in the Office of Research, Harris Lewin, Cam Carter, and Barry Klein, who are all here. They built this foundation that we stand on now. It's an incredible moment for UC Davis. More than ever, UC Davis is being recognized as an institution that is trusted to find solutions and just help make the world a better place. We're leveraging our scholarship to advance human and animal health worldwide, feed a growing population, adapt to a changing climate, and much, much more. And what distinguishes UC Davis is our focus on interdisciplinary collaboration. The solutions to complex, large-scale problems often lie at the nexus between disciplines. We all know that. We've heard that many times. And I think a lot of places claim that interdisciplinary research is one of their strengths. But having at least been to one other place and viewed many other places, I think it's really true here at Davis. Our greatest strength is our capacity to combine the talents of our researchers from multiple fields and explore those links between them. UC Davis is also unique in the sheer number of disciplines in which we excel. In fact, when I first came here, I was struck by the depth and breadth of excellence that's all across our campuses. It's not just agriculture and veterinary medicine, which have sort of the national or international reputation and world renown, but we're also leading in climate and sustainability research, clean transportation, life sciences, biological engineering, and on and on and on. Let me just mention a few of the accomplishments from this past year, and I can't mention them all, so just going to highlight a couple. UC Davis continued to play a critical role in responding to the COVID-19 pandemic by providing important insight into risk factors, transmission, social challenges, and treatments. We advanced human and animal health in many ways. For example, the All of Us research program received a $12.9 million grant from the National Institutes of Health to study the potential for improving health through precision nutrition. We developed new solutions to protect our planet and food supply, and that includes the work of a UC Davis team that identified a new gene variant in wheat that can increase the amount of grain produced. We also empowered a safer and more resilient society. For example, an interdisciplinary team of autism and general education experts is studying how to use inclusive math and literacy instructional practices to support learners with autism in K through 3 classrooms. UC Davis veterinarians developed an AI model to accurately predict life-threatening bacterial infection in dogs. UC Davis health researchers launched a novel study to identify a potential cure for HIV by using CAR T cells to target and control the virus without medication. Ermeus Krabrab and his colleagues in the College of Agriculture and Environmental Sciences are getting worldwide attention for their ingenious solution to reducing methane emissions from dairy cows by feeding them seaweed. And that research allows me to make many jokes. Finally, Delmar Larson in the Department of Chemistry developed an innovative solution to reduce the cost and environmental impact of textbooks. LibreText, an internet library of free and open source educational resources for K-12 and college courses, has been recognized around the world. And these, again, are just a few examples. There's more to come in the coming years, I'm sure. We recently initiated the UC Davis Grand Challenges Initiative to harness our expertise to solve some of society's greatest challenges. John Mazette, as you know, is leading this effort. Last month, as I mentioned earlier, we received the largest individual gift in UC Davis philanthropic history, a $50 million donation to create the Linda and Stewart Resnick Center for Agricultural Innovation to help drive a sustainable future in that field. We're also making progress in Aggie Square, which will provide new opportunities to leverage and expand our leading edge research and our commercialization efforts. So these are really exciting times, and I'm looking forward to building on all of these successes together with you. Again, thank you for everything you do on behalf of the university, and please enjoy the rest of the evening.
Thanks, Gary. Now it's my honor to invite Provost Croughan for her remarks.
So thank you, Prasant. Thank you, Chancellor May, and really, Prasant, just amazing. Congratulations on this accomplishment. I know it takes a team, and I'm going to name every single person involved. It will be done in about 2 hours, so just hang in there. But truly, I'm really delighted to join you today to just reflect on the collective expertise, the collective impact of UC Davis through our research, and to hear from our outstanding faculty. For those who don't know, most of this group in the front row is going to present on their research today. And I think I just got to call it out, Steve, if you don't mind. Steve Robinson and I were peer mentors together at the House when we were both undergraduates here more than 40 years ago. I will say I am the Provost and Executive Vice Chancellor now, but damn, Steve became an astronaut and distinguished professor, so what can I say? So I'm looking forward to hearing his research because that's one aspect I know less about. But I also really want to extend my heartfelt congratulations to you all on this incredible achievement. It's truly an occasion that we should all be celebrating. So UC Davis's continued growth in annual research funding, especially despite the constraints and challenges of the pandemic, or in part because of, I know there were faculty who took more time to write grants during the pandemic, but it has made us stronger and it's made us much more creative. It's incredibly impressive how our UC Davis research community leaned into the challenge of COVID to write more grants and to provide more solutions. One great example of how our university did this and how we led the way in research, it's our now wildly renowned COVID-19 testing program and our campus-led research contributions to the Healthy Davis and Healthy YOLO Together programs. The PCR and saliva testing modality we developed in our campus genome center was accomplished in just 19 days. I doubt there's very many of you who know that it was probably hour 2 that I was provost that I was contacted, I'm not joking, by Richard Mitchell-Moore and Ken Burtis and said, we think we can figure out a way to help the campus reopen. 19 days later, we had it figured out. That's UC Davis. That allowed us to provide rapid and accurate testing to not only our campus constituents and their families, but our entire surrounding community of Davis, and then expanded to all of Yolo County. We also, by the way, tested every athletic team that played against us. Once we were able to bring people back to the Mondavi Center, testing was provided again. It went far beyond just us. And as a result of this amazing endeavor, we were among the first universities in the world to be able to identify people when they were infected early on and still asymptomatic, and therefore prevent outbreaks and truly save lives. We didn't have a single case transmission on campus as a function of being at work. Community acquired, but nothing here. So as such, UC Davis provided one of the healthiest places in the world to both live and work during the pandemic. And all of this was only because of the amazing work that was done by our faculty, our staff, and our students. I would certainly agree with Chancellor May that the people of UC Davis truly are the driving force, not the equestrian reference it sounded like, behind our successful growth and impact. But as a Tier 1 research university, we are leading the way in a multitude of critically important and impactful ways. Reaching $1 billion in external research funding is a testament to the work being done on our campus by our faculty, our graduate students, our postdocs, our staff, and our undergraduates, and it's truly by everyone. So it is my honor to join you today to celebrate your work, your dedication, your contributions, and your commitment to UC Davis. I offer my most sincere gratitude to the entire research community, and I look forward to the exciting new developments I know are on the horizon and even more opportunities for innovative and transdisciplinary collaboration in the future. Go Ags!
Thanks, Mary. I'm truly delighted to invite a very special guest next, the UC System Vice President of Research and Innovation, Theresa Maldonado.
I'm the party crasher today. I just found out today that I was going to be here at the front. But I want to say first that I've been actually keeping an eye on UC Davis since the late 1980s. And why is that? Because one of my colleagues and dear friends, Andre Knosen, the department chair of electrical and computer engineering, he and I have known each other for much longer than I hate to admit in public, but we had the same PhD advisor, and so it's really great to see Andre today. I also know Chancellor May for many years, and it's a small world, and I'll talk about that in a minute. So $1 billion, extraordinary. And of course, the faculty and their students and postdocs are front and center. But who's behind the scenes? Research development officers, pre-award staff, post-award staff. You may know, you may not know, but these folks work nights and weekends to ensure that your proposals are submitted on time and that your awards are administered properly and in compliance. So I just want to do a shout out to research administration. Okay, faculty, so what's next? $1 billion. What's next? I'm watching. I want you to know that I work with Prasant very closely, along with his 9 other campus vice chancellors for research colleagues, as well as the 3 national labs, agriculture and natural resources, UC Health, and the natural reserve system. We meet all the time, and in fact, when COVID hit, that's when I started my job. I swear I didn't bring it from Texas, but I was literally shut down in a hotel when I first started this job. But Prasant, we met every single day on Zoom for the first 4 or 5 weeks to make sure research shut down properly, the animals were taken care of, and everyone was safe. And then we tried to figure out how to reopen as a system. It was extraordinary. I bring this up because this collegial group that has a lot of trust, the research leadership, came together to talk about what's in front of us, a climate challenge. And so I think we're in a period of time now that this is your next chapter. How do you pull together the expertise across the entire campus, including the policy folks, UC Health, Ag extraordinary, the vet school. But we're talking, this is how the $100 million climate money came about. Prasant and all of our colleagues in research leadership, we worked together late nights, weekends, pulling together a single system-wide proposal to show Sacramento that the University of California is ready to make an impact on the state, to vulnerable communities. And we need every one of you in this room to be part of that conversation. There are challenges to interdisciplinary research. I think we all have a passion for it, but how do we operationalize it? And we do need to come together. We are in interesting times, and we are going to do it as the University of California. So my office is working day and night to make this happen. And from policy to research peer review processes to innovation transfer. We're ready to work with you. I'm eager to strategize. We can do it with Prasant, with the deans, with faculty. I do have faculty contacting me. So I'm challenging you for chapter 2. Let's make a difference. And let's start with climate. Congratulations.
Thank you all for those very inspiring remarks. Now we would like to transition to our invited speakers for the evening. I just want to note here that we have many extraordinary, outstanding researchers on campus, and we could probably spend days here listening to many, many people speak. But today we have a sampling that we hope you'll enjoy. So we've asked each one of them to bring their most audacious ideas or visions for new research paths, untethered to expectations, and indeed to stimulate new thoughts, research, innovation, and collaborations. We also asked them to not bring slides. So we'll see how we do there. I am thrilled to introduce our first speaker, Professor Jan Nolta, from the Department of Cell Biology and Human Anatomy, and also the director of the Institute for Regenerative Cures.
Thank you so much. I'm absolutely thrilled to be here and to tell you a little bit about what we do. So imagine a future where, rather than medicating severe diseases and injuries, we could cure them. Imagine, rather than reaching for pills from the pharmacy, we reached for cells, living medicines, the building blocks of our body. And if those cells weren't functioning absolutely correctly, we could take them into the clean room in the laboratory and educate them to do their job better. That's what my team and I do every day, working toward curing different diseases. I started my career 30 years ago, not ashamed to admit it, 30 years ago, working on something called bubble baby disease. This is technically adenosine deaminase deficiency, if you want to get scientific. It's some of you may be old enough, as am I, to remember a movie called The Boy in the Plastic Bubble with John Travolta. That was about that disease. So the babies are born with no immune system. They need to stay basically in isolation. Their moms reach into them through plastic gloves. They have no immune system. They can't be outdoors or see other kids or even be hugged. 30 years ago, we started by taking the baby's cord blood stem cells as soon as they were born. The cord blood contains hematopoietic stem cells, these are the stem cells of the blood-forming system. Taking them into the laboratory, using a viral vector that was gutted and just contained the gene of interest, a good copy of the gene that was broken in the children's blood cells. Putting that gene into those cord blood stem cells, returning those to the babies. At first, it worked okay. And then we just kept at it for 15 years. And now, 50 children have been functionally cured. They're on no medication. They've had either their cord blood or bone marrow stem cells taken to a clean room facility. The good copy of the broken gene added to their stem cells, reinfused back into the children or young kids, and those stem cells make corrected blood cells, corrected immune cells for the rest of the lifetime of the children. And so we've had 10 years follow-up now on the most successful therapies, and we do call that a cure. Here at UC Davis, we're working on many variations of that therapy using people's bone marrow or mobilized peripheral blood. We can get the bone marrow stem cells to come out into the circulation and capture them through apheresis. We're very fortunate to have a large, good manufacturing practice facility at our institute in Sacramento. That's a large clean room facility where we bring the patient's blood or bone marrow cells or other types of cells. We bring them into the clean room facility and change them in some way. And so for those cells, blood-forming stem cells, we are treating things like sickle cell anemia. And again, after the cells are transplanted back into the patient, they will continue to make the corrected blood cells for the lifetime of the patient. So we're working on many cures with those type of stem cells. But I wanted to talk about one other type of therapy that we're doing. Imagine getting a diagnosis that you've relapsed from cancer was not as horrifying as it is right now. We are working with CAR T cells, which Chancellor May had mentioned for HIV. These are chimeric antigen receptor modified T cells. We take T lymphocytes from the patient's bloodstream, thanks to our apheresis in our cancer center. We bring them into the good manufacturing practice facility, and we add a gene that will put a new receptor, a new antenna on the surface of those cells. So I said that we can take the cells, the building blocks in, and modify them if they're not doing their job well enough. Well, the patient relapsed from their cancer because those T cells were not seeing the cancer. Somehow the cancer became invisible to their entire immune system. Cancers are good at hiding. We engineer the patient's own T cells to have a new receptor that we know will find that tumor, that cancer. We then take the cells back in our STEM mobile, which is a modified golf cart, take it back over to the cancer center, reinfuse them into the patients. They go through the patient's body and seek and destroy those cancers. So for leukemia and lymphoma, this is now up to 95% cure rate for relapsed patients. We're working on many solid tumors. This is the future. I'm so glad and humbled to be here at UC Davis, be able, for the last 15 years, be able to work toward therapies like this, make a real impact in our patients' lives, and to really use our robotic bioreactors in a good manufacturing practice facility to change people's cells to do a better job and not have to rely on all those pills in the pharmacy. I'm a 10-year cancer survivor, and this is very near and dear to my heart. As of last month, it was 10 years, so yay. Thank you. Thank you. Really, really glad to be here and just glad to help other patients. So that's all I have, very short, but thank you so much. Thank you for being here.
Thank you, Professor Nolta. Our next speaker will be Diana Farmer, Distinguished Professor and Pearl Stamp Stewart Endowed Chair, Chair of the UC Davis Department of Surgery, founder and co-director of the Department of Surgery Center for Surgical Bioengineering.
Well, thank you very much. It's really a pleasure to be here. So, you might say, why is some doctor here? Some MD doctor, unlike you official scientists, many of whom I see in the room and have been partners with. Really, I'm just a baby doctor. But I'm a baby doctor who's had the privilege of being at the intersection of two of what I think are the most exciting developments in my field in a long time. I had the privilege, I'm a fetal surgeon. I operate on babies before birth in the womb. It's actually not that new, even though it sounds pretty amazing. It's been going on for 30 years. And to take a line out of your playbook, just saying, UCSF thinks they invented it, but it really all happened here at UC Davis. Just saying. I was from UCSF, so I can say it. But it's true. The work was done by UCSF docs at the time, but at the large animal facility here in the Primate Center. Without that, it never would have happened. Most people don't know that story. But so I had the privilege of being at the start of just a pup in the early days of fetal surgery at UCSF. But I've also had the privilege of being now at the crossroads of taking fetal surgery and combining it with tissue engineering and really stem cell engineering. What brought me here from San Francisco, even farther than across the causeway, just saying, was the combination of that large animal facility that is an outgrowth of our fantastic veterinary school, and the fact that Jan's a little modest, if you haven't noticed that about her. The only GMP facility in Northern California at that time, and that one of only 2 in the whole state. We're a pretty big state. So little old Davis had our amazing GMP facility that has allowed so many of these things to happen. So I want you to think a minute. That's a fetus. And think about your grandparents having a baby. The fetus was shrouded in this veil of mystery. What did your grandparents talk about doing when a baby was born? Sort of counting the fingers and toes and making sure that everything was okay. Then along came an amazing disruptive technology, ultrasound. And all of a sudden, we could see inside. And oh my gosh, we saw all kinds of things going on in there. So we had to study the natural history of some of those things, because some of them went away. I mean, it was amazing. For the first time, we could see into the fetus. So as a pediatric surgeon who fixed problems after birth, we started to ask the question, well, we can fix the hole, it's what surgeons can do. We can cut and paste. But we couldn't necessarily change the pathophysiology that had already occurred. The problem happened before birth. So we started to just ask a simple question, very simple question. If we fixed it before birth, would that be better for the outcome than after birth? In the early days of fetal surgery, it was all fatal fetal defects because we had to balance. So here's a mom who has an operation for no benefit to herself, so undergoes all that risk. A little bit like transplantation, where you donate a kidney to somebody else. So it was similar. We had to balance the risk to her against the risk, the potential advantage to the fetus. So to balance that in the early days, we only addressed fatal fetal defects. So things that meant there'd either be in utero demise or the fetus wouldn't survive at birth. So along came, well, we learned that we could now actually do it pretty safely. A lot of it we learned at the Primate Center here. That's another whole story. But 10 or 20 years went by and we realized we could take care of these women quite safely, and we now treat tumors before birth, twins where one twin steals the other twin's blood supply, all kinds of things. It's amazing. So it's not quite so scary to get a diagnosis. There's things we can do. And right now I'm involved in what we call the CURE trial because we like Jan's approach to curing everything. So cellular in utero therapy, CURE, for spina bifida. That's the work some of you have heard about now. We have been excited to actually get FDA approval to take human placental-derived cells, put them into a fetus, first in human studies, all developed here at UC Davis, manufactured in Jan's GMP facility, animal pre-investigative work done here at UC Davis, and we've now treated our first 3 patients. While it's only a safety study, and we're only publicly reporting as a scientist those safety results, and I'm proud to say that we've passed our first safety trial, we're a little okay with the fact that the parents are all over social media. It kind of changes science now about how happy are that these kids that they thought would be paralyzed, these babies are wiggling their toes. So it is amazing. It may be a cure. I'm enough of a scientist. We're going to wait to get the rigorous data in a few more years, but in the meantime, we're going to let social media do its thing, as long as it's on our side. But I want you to imagine, what if, that's just one example, what if we could treat every potential disease that a baby might get before birth? What if we could cure sickle cell disease with just a little gene therapy and change that one gene that's responsible for that? Cystic fibrosis, tissue engineering for body parts that are missing. What if we could do all of that? And what if we could even tackle the fetal origins of adult disease? There's a lot of evidence that hypertension, that obesity, that many of the chronic diseases we have actually have fetal origins. And there's an intersection with our ag school and some of the exposures that we get from our environment. What if we could figure that out and cure all those diseases? Think of the money it would save taxpayers. Think of the lives it would save. Think of the families whose lives would be transformed, not having to, all the additional support that goes into these patients. What if we could do fetal gene therapy, fetal tissue engineering, fetal tolerance? If you knew you were going to need a transplant in the future, you could just set it up so that you wouldn't need immunosuppression. Fetal origins of adult disease, as I mentioned. This is not crazy. And half of those things I just mentioned are already done. And you are laughing at me because you think I'm crazy. I want you to know you're not the first. Nor probably will you be the last. People do think I'm a little overenthusiastic sometimes. Just saying. But that's not it. That's not all. So as a fetal surgeon, I was always amazed at the placenta, which everyone ignored and just threw away, right? This here is this most amazing organ, if you will. It doesn't even get credit for being an organ. Although they do make a stuffed animal, they make organ stuffed things. They do make a stuffed placenta. So I feel like I've made the organ list. I feel a little bit like Pluto. Now I realize there's an astronaut in the room. It's making me nervous now. I got another astronaut story coming. All right. Whew. Yeah, nobody competes with me with the placenta, although that's not true, actually. There's more competition of people studying the placenta now, and I like to think if I make no other contribution to science, it might be that I got people to study the placenta, because it is amazing. In 9 months, it becomes from nothing to an organ that makes blood vessels that are bigger than my fingers. It protects the fetus from being rejected by the mother, so it's an amazing immune organ in its own right. It's an endocrine organ. It provides all the nutrients, and it figures out how to do that, and it supports that growing fetus. And then it becomes senescent. Then it just stops. So I want all my vascular scientists to study the placenta. I want everybody who's interested in aging to study the placenta, because Aijin and I, my scientific partner out there, we do not have time to do it all. So please get studying the placenta. But imagine if we could harness, if we could really understand those things that the placenta can teach us, and imagine if we could really understand all that amazing biology that makes fingers and eyeballs and fingernails and hair and hearts and bones that happens in a developing fetus. So really understanding developmental biology, having it be more than a descriptive study, would give us profound insight into ways we could help improve human health, solve problems. I think there's no more exciting time to be a scientist, and I don't think these things are crazy in terms of trying to solve them. It requires our academic medical centers, and even better, academic medical centers that are part of a bigger university. Just saying, poor UCSF's just out there by itself. The interdisciplinary work that can be done with our ag school. I want to grow organs in our ag school for transplant. I want to talk to our pig people so we can figure this out. It's already being done, but Davis should be leading that. I know that sounds scary to some of our veterinary colleagues, but face it, unless you're a vegetarian, which, by the way, is not a bad idea, many animals serve important needs for humans, so I'll just stop with that. But are you all aware of the DART project? This is where I get back to my hands up. Almost everybody. Okay, so I don't really understand. My husband was an astrophysicist by training before he switched into medicine, but I think of this as somebody basically sat in their backyard, shot a BB gun to a place where the moon was going to be 5 years from now, and hit a Coke can on the moon. I mean, that's the only way I can figure out. I mean, how crazy is that? That you could shoot a little tiny thing and hit a Coke can on the moon where the moon was going to be 5 years from now. But I think that's what they just did. So if they can do that, I don't think there's any problem in solving all the diseases before birth. Thanks, you guys.
That was fabulous. Thank you, Dr. Farmer. Next, we will hear from David Block. He's a professor and Marvin Sands Department Chair, Department of Viticulture and Enology, and the Ernest Gallo Endowed Chair in Viticulture and Enology.
Thank you, and thank you for having me here. We're going to move a little bit from medicine to food here, and I want to start by having everybody imagine a perfectly marbled steak. Now, imagine if you didn't need a cow to make that steak, a real steak. And I think, how would that work? Well, this is the idea that's a relatively new field of cultivated meat. In cultivated meat, you would isolate stem cells from an animal, say a cow, and you take those stem cells and you'd grow them up in a lab and media and nutrients that would allow those cells to grow. And when they grew enough, you would take all those cells and you put them in a slightly larger container and grow them up there and keep on going step by step until you got to full production scale, which is likely to be something like a 250,000 liter stainless steel fermenter. How big is that? Well, that kind of fermenter would fit about from where I am to the wall and be taller than this room. It's huge, growing cells. And you'd grow those stem cells to that size, and then at that point, you'd have to do something to differentiate them into all the parts of meat: muscle, fat, and connective tissue. Right? And you could take all those cells and mush them back together into a hamburger, or maybe better yet, 3D print them into that perfectly marbled steak. Well, why is this idea important? Well, there's lots of reasons why this idea is important, but perhaps the most pressing of those ideas is the fact that in the next 30 years or so, the population of Earth is going to grow to between 9 and 10 billion people. In addition to that, in developing countries, people are adding more and more meat to their diet, and they want to do that. Well, this means, by our estimates from here and other places, that by 2050, we're going to need to produce 60% more meat for people's diets in order to satisfy those dietary needs. Well, that's a lot. There's already more than 300 million tons of meat consumed on Earth every year, And 60% more of that is a huge amount. And how is that going to happen? Well, I suppose one possibility that comes to mind is you just grow more animals, more cows, more pigs, more chicken, and so forth. But experts tell us that that's just not going to work. There's not enough resources on Earth to grow more animals to fill that need. And so there has to be other ways of doing it. Cultivated meat would be one of them. Now, there are other reasons as well for wanting to work on cultivated meat and to get that to work. Things like potentially more sustainability or worrying about antibiotic resistance that might arise from growing more animals, or animal welfare, obviously. And I think one that we don't often think about, but we could, is trying to even out natural agricultural cycles that happen when farmers or cattlemen raise cows for meat or any other kind of animal for meat. They go through ups and downs based on the weather and the climate change, and it's possible that the same raw materials could be used for cultivated meat as for growing those animals, produced in the same location or very nearby, and using the same distribution channels to get that meat to market, thus evening out those ag cycles. So there are lots of reasons to be thinking about cultivated meat. And the question is, if there are that many reasons to think about it, if it's so important, why isn't cultivated meat on the market yet? Well, there are about 100-something companies in the world that are working on trying to get that cultivated meat to market. And in fact, I didn't tell exactly the truth because there is one product, cultivated meat product on the market. It's a chicken nugget. It's actually produced by a Northern California company, but in Singapore. And it's served in 1 or 2 restaurants in Singapore where you can buy these cultivated meat chicken nuggets for about $15 a nugget. Well, this is not a high throughput solution to world hunger, right? So why is that? Why hasn't anybody been able to do this at scale yet? Well, there are 4 major hurdles that we have to get over to be able to produce cultivated meat at scale. The first one is we need cell lines that grow when you need them to grow and differentiate into fat, muscle, and connective tissue when you need them to differentiate. And because those things are sometimes diametrically opposed, it's not an easy problem. The second hurdle that we need to overcome is we need inexpensive media, nutrients, to grow these cells on. Because right now, for growing animal cells, people look to the pharmaceutical industry where they're selling products for $10,000 a gram. It's just not going to work for food. So we have to come up with probably plant-based or microbe-based media that are going to be really cheap and support growth, and not only support growth of these cells, because this is a food, the finished product has to taste good, it has to be healthy and safe and nutritious and stable, and all these things that the media are going to help the cells do. The third hurdle is creating structure, because it's one thing to create a hamburger, and it's way more complicated to create a marbled steak or a salmon fillet or a chicken breast that has the right texture, the right cooking properties, the right nutrition, all that stuff. Way more difficult. And then the fourth hurdle is, well, companies have gone and spent 2 weeks making a piece of salmon, sashimi-grade salmon. It's obviously not commercial that way. What we really need to do is make one of those pieces of salmon every second, or more likely hundreds or thousands of pieces per second, to be able to have this fill the need that the Earth has for more dietary protein. Now, luckily, UC Davis is unique in that we have pretty much all the disciplines on this one campus to get through those hurdles, solve these problems, and get cultivated meat to market. That includes people who are muscle physiologists, tissue engineers, stem cell biologists, animal scientists, food scientists, chemical engineers, bioprocess engineers, bioengineers, food scientists, sensory scientists, ag econ people. All these people are needed to solve these problems, and we're doing that here at UC Davis now. We have the UC Davis Cultivated Meat Consortium that started just a couple years ago. As of 2 years ago, we had about 4 or 5 people working on this on campus, and now, just 2 years later, we have over 50 researchers on campus working on solving these hurdles. This includes 20 faculty members in 8 departments over 4 colleges and schools, and students who are passionate about getting these products to market to solve these huge problems. And I would say we've made good research progress already. We're teaching students that have already gone out into this industry. A lot of these companies are in Northern California, which is great. We've started continuing education programs to help these companies grow. And this is something that UC Davis does really well, getting ag and food industries to grow. Think about almonds. Think about pistachios. Think about wine. I'm in the wine department. I should at least mention that while I'm here. Think about leafy vegetables. These industries all grew because UC Davis was there to help them grow. And I think we're going to be doing that for a long time for cultivated meat as well. So I just want to finish by saying I've been talking about products that mimic what's already on the market: conventional meat, steak, chicken breast, salmon fillet. And in addition to cultivated meat, where we're trying to make those products, which I think is a good idea from consumer acceptance point of view at first, there are plant-based meat products and there are fungal-based meat products that are on the market already. I think one of the really cool things that's going to happen in the next generation is to not just be mimicking new meat products, but creating new ones that are even more delicious. They're healthier. They have more nutrition. They're safer. They're more stable. They can be grown in different places and they're cost effective as well. And I think this idea of these hybrid products is what's really exciting moving forward. There will be foods in the next generation that we've never seen in our lifetime. And I think that they'll probably come from the kind of passionate food scientists and other scientists that are working on this project who will someday come to us and say, you know what? I have this out-of-the-box idea for a new idea. Imagine if. Thank you.
Very interesting. Looking forward to tasting all these new protein products. Next, we have Dr. Priya Shah. She's an assistant professor in the Department of Chemical Engineering and the Department of Microbiology and Molecular Genetics. Priya.
All right. Well, first of all, thank you very much for the invitation, Prasant and Chancellor May. It's really an honor to be here and contribute my small portion to that $1 billion of research funding. So to start, I want you to think about Ebola hemorrhagic fever, AIDS, West Nile encephalitis, SARS, bird flu, congenital Zika syndrome, and we can't forget COVID-19. All of these diseases are caused by viruses that emerged by jumping into humans from another animal. And so if this list of diseases doesn't underline the importance of studying viruses, even if they don't cause disease in humans yet, I want you to imagine Ebola-like hemorrhagic fever of unknown origin, a new Zika-like virus that causes birth defects, or a novel coronavirus that causes COVID-32 10 years from now. The virus that causes the next major outbreak is already out there. But imagine if we could actually develop smarter antiviral therapies to be prepared for that next outbreak. Imagine if we could do this in a way that didn't result in drug resistance to these antiviral therapies, the way we have to worry about drug resistance for HIV or influenza virus, or potentially even SARS-CoV-2 with Paxlovid. Now let's get really wild. Imagine if we could have smarter antiviral therapies that would work without even knowing which virus is going to cause the next pandemic. Then we would be truly prepared. And I think that we can accomplish all of that. But to do so, we really have to build generalizable themes about all of these unrelated viruses, but with molecular level resolution. So to explain why we think that molecular details of virus replication are so valuable to combating viral infections, what I want you to do is imagine a little more. Imagine a single cell in a human body and think about what a virus needs to do inside that cell to be successful. I'll argue that it has to do three major things. It has to get into the cell, it has to make more of itself, and then all of those new viruses have to get out. And so viruses have to do all of this with a very small set of viral machinery. And so compared to the host, which has a much larger set of molecular machinery, viruses have started to hijack the molecular machinery of the host. Viruses are master manipulators of cells, and they use the molecular machinery of cells to turn cells into tiny little viral factories. And so instead of targeting the viral molecular machinery with our antiviral therapy, imagine if we instead attacked the host molecular machinery. And this is exactly what my team of chemical engineers and biologists really think about when we do all of our experiments. So my team builds new systems to study these molecular events of virus replication with quantitative detail. And I could talk for hours about how we do all of this work, but what I really want to do today is tell you about why this work is so important and why it's powerful through the lens of building smarter antiviral therapies. One of the types of molecular events that my group really loves to study are protein interactions between a viral protein and a host protein. So sometimes these protein interactions help the virus replicate. Sometimes these protein interactions actually help the host defend itself against the virus. Sometimes these protein interactions actually cause disease on a macroscopic scale, like we've discovered with the birth defects that are caused by Zika virus infection. And so how does studying these molecular interactions actually help us build smarter therapies? Well, if you think about how your traditional antiviral drug is developed, it's developed by targeting a viral protein. And so the drug has to really fit really nicely with the shape of the viral protein to be effective. Slight changes in the shape of that viral protein could render that drug completely useless. These differences in shape could be because you're trying to use the drug on one virus, even though it was designed for a completely different virus. So we've got Paxlovid now, but what if there's a novel coronavirus 10 years from now? Will Paxlovid still work? Will that viral protein still look close enough to SARS-CoV-2's viral protein? We don't know. The other way these differences in protein shape can come about is through the actual mutation process that occurs during virus replication. And that's exactly what we see with drug-resistant strains of HIV or other viruses. So now let's flip this around. Let's actually target the host machinery instead. One of the advantages of targeting the host machinery is that now mutations in the viral proteins don't really matter anymore. The drug will still bind the host machinery quite well. And so for a virus to really develop resistance to a host-targeted therapy, the virus would have to actually find a replacement host target. That's a much more difficult problem for the virus to solve. And so this therapy effectively traps the virus where it is. Another exciting advantage of a host-targeted therapy is that actually a lot of unrelated viruses will still use the same host machinery to replicate themselves. And so if you can identify that machinery across many different viruses, you could potentially have a drug that works against SARS-CoV-2 and Ebola virus and Zika virus. And so differences in the viral protein shape don't really matter anymore. What are the implications of this? Well, let's suppose we had a drug that was active against SARS-CoV-2, Ebola virus, and Zika virus. Well, there's a high probability that that host-targeted therapy would also be active against a novel coronavirus that we identify 10 years from now, or a new Zika-like virus that causes birth defects. And so this is just one way we think that we can build smarter antiviral therapies through targeting virus-host protein interactions. Now, what's stopping us from this being a reality? Well, first of all, we just need more data. There is such a rich world of viruses out there that we know very little about. We don't understand how they interact at the molecular level with the host machinery. So we can start to fill these gaps in knowledge with unbiased, discovery-based research. And we can focus our efforts on virus families that have a high risk for emergence and new outbreaks. The tricky part, though, is that federal funding agencies don't always fund this type of discovery science or fishing expeditions to the level that you would need to support the type of vision I just described. So we really do need to find new ways to support this type of discovery research. The other thing that we need to do is build new partnerships. We need to identify collaborators with complementary skill sets that can help us identify the virus families that pose the highest risk, that can test the host-targeted therapies once we identify all of those host targets. So all of that capability actually exists here on UC Davis campus, which is really great for me because this is my vision. But these are just a few of the pieces of the puzzle that we need to lay down to make this a reality. But it's a start for building smarter antiviral therapies. Thank you.
That was fantastic. I would like to invite our next speaker to the stage, Isabel Montañez, Distinguished Professor in the Department of Earth and Planetary Sciences and Director of the Institute of the Environment.
Is it on? Oh, it is on. Making life complicated, I have a prop. So, 416, what does that number mean to you? I invite you to think about a large or giant glass container filled with 1 million marbles, of which all are blue, except for 416, which are red. Would you even notice those 416 marbles? Why not, right? But here's the thing. Those 416 red marbles are wreaking havoc in our climate system. And they're presenting us with the biggest challenge of our century, climate change. 416 is currently, in parts per million, in the air that we breathe, the amount of carbon dioxide. And that is the primary greenhouse gas behind our global warming. Using geochemical techniques like those that we've developed in my lab over the last couple of decades, we know that CO2 in the atmosphere has never been this high since humans have evolved. In fact, it hasn't been that high for 3 million years. And in those 3 million years, the Earth has experienced repeated extreme climate changes. In California, these are anomalous climate or weather events like century-long mega droughts and mega floods that made the Central Valley into a giant lake. All anomalous events, but naturally driven at the margins of the climate system. But what really is the take-home point is all with 100 to 200 fewer red marbles in that container. So how do we know all of this about 3 million years ago and up even to today. We know it from archives of crystallized climate, like this stalagmite that came from a Sierran foothill just an hour or so from here. And if you can see the layers, there are actually thousands of laminations in here, and entombed in each of those is fossil rainwater that was trapped when the mineral layers were laid down each year. And just like the mosquito trapped in amber from Jurassic Park, that has that genetic code of the dinosaur in the fossilized blood, the fossil water in this stalagmite has its own genetic footprint that we can tease out using high resolution instruments. And from the chemical information in these layers, we can extract evidence that are major changes in temperature, in rainfall, in wildfire activity in the past, in those past 3 million years, often in sync with major climate change. So today, with up to 200 fewer red marbles in that giant container, humans are supercharging our climate. And thus, any future extreme climate events will most certainly be of greater magnitude than those of the past, archived like in this stalagmite. So that said, I can say I'm hopeful. I'm actually very hopeful. And you should be thinking, why? Because I can imagine a future where we learn to manage our planetary climate, and where climate change is no longer an existential threat. You may not realize, but we are actually in a revolution right now. We're starting to invest trillions in climate mitigation, climate adaptation. The US is leading the way with a recent pledge to reach net zero carbon emissions by mid-century or sooner. And this goal is going to require massive decarbonization in the next couple of decades. And right here at UC Davis, we have leaders who will be involved in the innovation and the implementation of new technologies that no doubt will shift us from the fossil fuel consuming society we are. But my message is that's not going to be enough. We're going to need other ways to decarbonize our world. So imagine if silently in the background, while we develop these technologies, we exploit one of Earth's longest lived and most powerful natural reactions, capable of sequestering billions of tons of CO2 per year, and thus being a part of our decarbonization portfolio. Since this planet has had water, there is a chemical reaction between water and rock that sweeps CO2 from the atmosphere and locks it away in minerals below the Earth's surface. Actually, what happens, rain pulls that CO2 out, it makes carbonic acid, it infiltrates soils and rocks, it dissolves them, and it makes inert carbon that is then sequestered in new minerals for thousands to millions of years. But this natural process is slow. It's on geologic timescales. So at the Institute of the Environment, we see the potential to accelerate that natural process to rates relevant to climate change. How so? We call it enhanced mineral weathering, but it involves adding ground minerals and other amendments like kelp, wherever Ermias is, combining these amendments, adding them to agricultural and rangeland soils, and ultimately to all that land that we anticipate will be decommissioned with climate change. We envision next generation monitoring, coupling it with precision irrigation, where we can define new soil amendment strategies that are tailored to the individual land manager's needs, all while sequestering CO2 from the atmosphere and improving the water and air quality for many rural and underserved communities. So this idea that we can harness the sequestration potential of one of Earth's longest known processes or reactions with minimal new technology and we can actually remove a good portion of those red marbles from that giant container is, for me, reason to be hopeful. Thank you.
Thank you, Isabel. Next, we will hear from Karen Moxon, professor in the Department of Biomedical Engineering.
So I don't know about you guys, but my head is spinning with all the possible things that could happen here. It's just quite amazing. So today I'd like to talk to you a little bit about what's going on at the UC Davis Center for Neuroengineering and Medicine. And so I can kind of tailor what I'm going to say here. How many people have heard of neuroengineering before? Oh, that's pretty good. Okay, I'm excited. So then, so brain-machine interfaces? And how about Elon Musk's company, Neuralink? Pretty much everybody. Okay, that's great. That's great. It's going to make it so much easier for me. So the field of neuroengineering has really exploded in about the last 30 years. In this field, what we can do is for paralyzed patients, we can actually implant microelectrodes into their brain tissue. So we take their skull off and actually put those microelectrodes into their brain tissue. We record those neural signals, and then we decode their intention to move, and then use that signal to control some sort of external device, like a robot arm or your cursor on a computer screen. And while this technology is pretty amazing, there's a lot of work that needs to be done to make this a common technology for routine patients, hence why Elon Musk has his company Neuralink that goes in fits and starts. But there's other companies that are interested in this as well, like Meta and the car company Nissan. Here, they want to decode your brain state. So are you paying attention so they know when to slip you that important advertisement? Or are you going to fall asleep in your car so that they can send you a signal so that you can pull over to the side of the road and avoid a crash? But what if we wanted to restore movement, and rather than just control some external device, we wanted to enable that person to actually get up and move? So here we imagine not going and controlling some external device, but using that signal to control, let's say, an exoskeleton. So if you're paraplegic, we could have an exoskeleton and use those signals to enable you to stand up and walk across the room. If we can think of this, then you can imagine, what about those people who work in high-risk environments, like firefighters and soldiers who have to carry around these huge, big backpacks? Well, what if we could enable an exoskeleton for them that they could just think about, and then they could continue to use their hands for the tools that they need to maneuver while effortlessly picking up these heavy loads and maneuvering them around? And the value of advancing these technologies is more than in just the end result, because what we learn from the data that we collect, and let's face it, the failures that we encounter as we try to develop these technologies, that's going to teach us more about how the brain encodes motor information and how the brain functions. And then that, in turn, allows us to develop even better technologies, which then gives us even more insight into how the brain works. And this gives us that great, wonderful, virtuous cycle that we all like to be a part of. So what if instead of having this person have to carry around all this exoskeleton, we can actually have them move their own legs under their own will? So now imagine we're taking the signal out of the brain and using it to stimulate, let's say, in a spinal cord injured patient below the level of the lesion. This is what I do in my lab, and I'm collaborating with several people here at UC Davis campus. But what does this require to make this happen? Well, it requires us to be able to decode those neural signals. And okay, we've been doing that pretty well for the last several years, so that part, needs more work, but it's going okay. But in addition, we need to develop novel control systems that can take this decoded signal and determine exactly how to stimulate. And we need novel neuromodulation systems that can precisely stimulate into the nervous system to restore exactly the type of locomotion or movement of the limbs that people desire. So again, in the theme of, I think, what we've all been talking about here, this is big team-based science. And we're so lucky to be here at UC Davis, where, as Dr. May said earlier, people are really excited about these types of interdisciplinary research. So I could talk about dozens of people, but I'll just highlight a couple. So for example, there's Dr. Croughan in the Department of Mechanical and Aerospace Engineering, who's an expert in control systems and really interested in taking this knowledge and applying it to neural systems. And then there's Dr. Erkan Shekhar, who I actually think is here today in the Department of Electrical and Computer Engineering, who's developing the most advanced neural interface systems that can not only record neural activity like Musk's Neuralink company, but can also stimulate back into the neural system that we would need for this type of a device. And there's lots of other faculty that I could talk about. So now, okay, let's take it to the final next level. Imagine if we were all outfitted with something like this. So now forget Siri. We can just think about controlling the lights, turning them on and off, controlling our heating and cooling systems, controlling our phones and our computers. I don't know about you, but my hands definitely get tired after all that typing. And I get this pain in my shoulder from having to control the mouse. And if I could just make the words come out on the page as I want them to, as I think about them, it would be great. But of course, here we get to, just immense ethical and social problems that evolve. And I guess I just like to say that that's one of the advantages of having this great interdisciplinary center like the Center for Neuroengineering and Medicine, because in addition to moving these technologies forward, these are topics that we care deeply about, and we bring together social scientists and ethicists and engineers and clinical investigators to try to address all of these problems together so that as we move these technologies forward, they can be really the greatest impact and the most benefit to society. So I hope you've enjoyed this little slice of what's going on at the Center of Neuroengineering and Medicine and take time maybe to look into what's going on there. So thank you very much.
Thank you, Karen. Our next speaker was unable to attend today, but recorded his talk for us. I would like to introduce Dr. David Olson, Associate Professor in the Department of Chemistry and the Department of Biochemistry and Molecular Medicine, and the Director of the Institute for Psychedelics and Neurotherapeutics.
Nearly 1 in 5 people will experience a mental health condition at some point in their lifetime. Many more will be ensnared by the grips of dementia as they age. Finding ways to promote brain health is one of the grand challenges facing society today. Billions of people worldwide have diseases like depression, schizophrenia, post-traumatic stress disorder, Parkinson's disease, anxiety, substance use disorder, Alzheimer's disease, and our current solutions are far from ideal. Many conditions that impact how we think, how we feel, and how we behave are caused by physical changes in the structure of the brain. Neurons atrophy and lose the ability to communicate with their neighbors. But the current tools we have to combat many of these illnesses don't directly address these broken neural circuits. Right now, the best modern medicine can do is to provide treatments that need to be administered daily to mitigate disease symptoms, medicines that often come with a number of undesired side effects. But imagine if we could give the brain the means to heal itself. What if we could repair those broken neural circuits to achieve long-lasting remission of disease symptoms? And what if that meant only taking a pill once a week, once a month, or even once a year instead of every single day? At the UC Davis Institute for Psychedelics and Neurotherapeutics, we are working hard to transform that idea into a reality. Psychedelics and related compounds like LSD, psilocybin, and MDMA have demonstrated enormous potential for healing damaged neural circuitry and producing long-lasting therapeutic effects across a wide range of indications. Their ability to promote neuroplasticity is opening new doors for improving brain health. Despite their promise, these compounds are not without risks, and challenges remain regarding their effective integration into our current healthcare ecosystem. To ensure patient safety, these powerful substances must be administered under the supervision of a healthcare professional, which drastically increases the cost and complexity associated with the treatment. Furthermore, some patients cannot take psychedelics if they have certain diseases like schizophrenia or a family history of related illnesses, and others may not want a psychedelic experience for personal or religious reasons. But imagine if we could separate the hallucinogenic or intoxicating effects of the drugs from their ability to promote neuroplasticity and heal damaged neural circuits. If we could achieve this, we could help many more of the billions of people worldwide looking for new solutions to improve their mental health. This possibility may become a reality sooner than you think. Researchers who formed the UC Davis Institute for Psychedelics and Neurotherapeutics have already made incredible progress towards this goal. Using the tools of chemistry, molecular biology, and modern neuroscience, we have engineered the first non-hallucinogenic variants of psychedelics capable of producing long-lasting therapeutic effects in models of depression, post-traumatic stress disorder, substance use disorder, and schizophrenia, among others. Next, we plan to expand our efforts to identify neuroplasticity-promoting compounds for the treatment of neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Treating a disease is one thing, but what if we could prevent it altogether? By fully understanding how psychedelics impact brain structure and function, we hope to develop new strategies for making the brain more resilient. Imagine if there was a pill that first responders or military personnel could take to prevent the onset of post-traumatic stress disorder. What if there was a prophylactic that could make you immune to the depression-inducing effects of chronic stress? If we can find ways to optimize brain health, maybe we can stave off dementia as we age and extend our health spans. To accomplish these lofty goals, we will need to embrace bold ideas and not be afraid to venture into areas once considered taboo. We will need the support of visionary philanthropists, foundations, and government agencies. We will have to build bridges between intrepid scientists from a variety of disciplines, and we will need to forge partnerships with industry to ensure that academic innovation ultimately ends up in the medicine cabinets of patients. What we are hoping to achieve will be no small feat, but we hope you will join us in our efforts to find solutions to some of the greatest challenges in neuroscience drug discovery.
It is my pleasure to introduce our next speaker, Maria Marco, professor in the Department of Food Science and Technology and chair of the Food Science Graduate Group.
Hello. So when I was a child, I was fascinated with outer space. I recall going to my grandparents' house and turning on the TV, yes, like that, to watch Cosmos with Carl Sagan, and going to the library and scouring through books on facts and figures about the solar system and the universe. Why did I do this? I was fascinated. Could this even exist? So far away. It was mystical. But I'm not here to tell you about the virtue and importance of outer space. We'll have another great speaker to follow to do that. No, I'm here to tell you about an equally mystical and expansive world of microorganisms. But unlike celestial bodies, microbes are here on us right now, in this room, on our hands, on our chairs, in the air, and we could all say, yuck. And I think we're owed that after the past few horrible years of our pandemic, just underscoring the importance of these microorganisms, how one virus could change our lives so dramatically. But don't worry, most of the microbes in this microbial world do not cause us harm. In fact, they do good. Just as telescopes and satellites have transformed how we see outer space, so too have the technological advances of the past 30 years revised our version, our understanding of microorganisms. To give you a little bit of a picture of what we found, we now know the vast genetic diversity of microorganisms on our planet. We can only grow in the lab and understand them less than 1%. And what this means in the genetic diversity and the functional potential is that we look just like our pets, cats and dogs, compared to the expansive functional differences of microorganisms. Let's talk about numbers. It's estimated there's 200 billion trillion stars in the universe. Well, there's 1 million more bacteria on the planet right now, 10 times more viruses than bacteria. We have all the other microbes. We really, truly are outnumbered. So then it's not so surprising when I tell you how important these microbes are. 60% of our Earth's biomass is microbial. They're important to grow all of our crops, all of the plants. Clean air, clean water rely on microorganisms. Our own bodies are filled with trillions of them right now. There's more bacteria in our digestive tract right now in each of us than people ever lived on Earth, so the total of human population. What are these microbes doing? They're digesting that food we just ate and our drinks, giving us energy, protecting us against pathogens, even signaling to our brains, changing our behavior, our moods. They're important. But for me, the most tangible way I can think of microbes in this microbial universe is through foods. Of course, we've seen rotting fruits and vegetables or had foodborne illness, but there's much more to microbes than that. Microbes make the food we eat. They make fermented foods. Fermented foods are just that, just like this beautiful plate of sauerkraut, kimchi, bread, beer, wine, yogurt, cheese, and let's not forget coffee and chocolate are only around because of the microbes that make them. And beyond this, they're really important for our culture. They take us back to our earliest humans and are propagated by all cultures around the world today, even though may not know what a microbe is. So with all of this power of microbes, I'm going to give you a few imagine ifs. Imagine if we could see the unseen and understand our microbial world. Imagine if we had a device that we could attach to our phones and take a little bit of saliva or a swab, and within minutes tell us what microbes we have or maybe we don't have and need or their enzymatic potentials. We could take this to our pharmacist or physician, and they'll prescribe us or tell us which microbes we should take. Our lab and others have already found that certain microbes can really change our health. They can help keep off the pounds. They can also repair damaged intestinal lining, improving gut health, and many other ways. But we're really just at the cusp of understanding this as we explore the microbes on our body we know as the microbiome. Imagine if we could stop the spread of antimicrobial resistance. We're on track within 30 years to have more people die of bacteria that are resistant to antibiotics than people die of cancer. We need to find solutions quick. So to do this, myself and a team of an interdisciplinary team internationally, we're looking at addressing this for farm animals. They use more antibiotics each year than we receive as people. And if we could do this by building a better microbiome from birth, for people and for animals, we'll reduce the need for antibiotics and therefore antibiotic resistance spread. Another imagine if is imagine if we could use the 1 billion tons of food waste we throw away every year. Instead of putting it into the landfill, we use microbes, like the microbes used to make sauerkraut, to give us new products, new fermented foods, new flavors, changing not only the safety of our food supply, but also the sustainability in our planet. And lastly, imagine if we not only had NASA, but we also had the National Microbes and Humanity Administration, bringing together scientists like microbiologists like myself, scientists of all disciplines, but also experts in humanities and arts, the public and lawmakers to take a radically different view of our microbial world. And I'm so proud to be saying I'm doing this here with others in the humanities and elsewhere to take another look at fermented foods. And instead of just extract the microbes, but to work with the people who've made these foods for centuries, out of respect and understanding of the many different ways that we can look at microorganisms. So with this, I'm going to leave you with a saying that was attributed to Carl Sagan, and that is, somewhere, something incredible is waiting to be found. Thank you.
Thank you, Maria. I was once a microbiologist, so that hit me in the heart. And last but not least, I would like to introduce Stephen Robinson, professor in the Department of Mechanical and Aerospace Engineering, the director of the UC Davis Center for Spaceflight Research, and director of the NASA Habitats Optimized for Missions of Exploration Space Technology Research Institute, which was one of the first projects I worked on when I came to UC Davis. So I was very excited to hear you talk.
Thank you, Prasant especially, for inviting me to this amazing, me and my microbes, to this amazing celebration. So imagine a huge rocket that is capable of lofting satellites 23,000 miles above the Earth to look back at the Earth and to look back at what we are doing to our planet, our environment, our atmosphere, and even us. Land masses, water, oceans, temperatures. Imagine this huge rocket. Is 3 rockets strapped together. There's 27 rocket engines. They all have to work perfectly. Imagine this launch. 2 of those rockets finish lofting, getting the rocket up to speed. 2 of those rockets actually turn around and fly back and land back on Earth to be used again, while the middle rocket goes off and brings the payloads up to orbit. When will that happen? The last time it happened was this morning. Now, imagine the largest rocket built by the United States, larger than the Saturn V that took the Apollo missions to the moon. To be able to go to the moon, where to learn about things we didn't know in the 60s, there's water there. And eventually to Mars, there's water there. Where water is, we can do many things. Humans can do many things. The point is not to go to space to develop these technologies to use in space, but to learn things and develop toolboxes and to teach students, our graduate students and our undergraduates and ourselves, about how to make this life more survivable, more in tune with the environment, the one environment that we inherited, and to learn to live beyond the threats that we are generating ourselves. This is what the space program is for. So imagine this huge rocket able to go to the moon. It has giant rocket engines. It has 9 huge rocket engines, and it's going to go 243,000 miles to the moon and return with a capsule that can carry humans later, return to the Earth. When will this happen? In 13 days. 13 days from now. Now, imagine the biggest rocket ever built by the human race, able to carry vast amounts of mass and humanity into space, to learn about humanity in space, physiology, people in space, to be able to loft bases to the lunar surface so we can learn yet more about these things that we have such a hunger and need to learn about. This rocket is a monster. It has, in the first stage alone, 36 rocket engines, each of 500,000 pounds of thrust. This is science fiction. When will this happen? By the end of this year, the SpaceX Starship. And imagine people living in space for 22 years. Crews swapping in and out on a spaceship that has international crews coming from many, many backgrounds over 22 years. Every single astronaut and crew member came back safe and alive and healthy to learn about what happens to humans in very hazardous environments, to learn about how to extract carbon dioxide from the air, to learn about how to reuse the water that we have already used, to learn about the kind of things that we need here. Imagine that doing that for 22 years. When will that happen? Well, year 23 of that on the International Space Station started this week. That's just today. Let's talk about the future. So imagine you are in space, and you have been for 4 months on the way to Mars from lunar orbit. And as simple as that sounds, well, It doesn't sound very simple to me, but actually there's a lot of things that we don't know about that. What are the long-term health effects on your body? Zero gravity affects many aspects of your physiology, much like aging here on Earth. Long duration exposure to low-dose radiation. We have much to learn about that. Influences of fluid shift on your ocular properties, your renal system, and of course your musculoskeletal and vestibular system. So much to learn. What are the mental health aspects and the challenges from long duration isolation? Imagine being away from Earth for 3 years. You know, one thing about being in space, I can tell you, is the fact that you can see the Earth brings mental health into your being. And as soon as you are beyond, you know, when the Earth is just a dot, when we look up, when we see Mars, you know, you have to look hard, you have to know where it is. And if you saw Earth like that, it would not bring that sense of home back to you. What kind of countermeasures for these challenges? Now, I'm talking engineering, pharmaceutical, behavioral, and so on, can we develop that will benefit you, the space explorer, but more importantly, us, the everyday humans on Earth? So imagine that you share control of your spacecraft with an AI-enhanced control system that are learning all the time to monitor the subsystems of your habitat, the thing you're depending on to live, predict future failures, and build new parts as needed through additive manufacturing. How will you understand what the AI systems are doing, and how will you know when to intercede? Imagine if you have a team of robots on board to assist you and your crewmates in maintaining the spacecraft that your life depends on, reconfiguring it as things change, and repairing things as needed. How much can and should the robots do versus you and your fellow humans? Should you work together with the robots, share responsibilities, or should you have direct command of what the robots do? How can you remain safe when the robots are moving under their own autonomous control? How will you understand what they intend to do? And then, when surprise failures occur, and let me tell you they do in space, how will you or the onboard autonomous systems know what to do next? This is where humans really come in. What skills must you have before you launch to ensure that you're ready for anything that happens? And then, how do you say that about the artificial intelligence systems? Are they ready for safety critical surprises? Because it's going to happen. How will you know if they aren't? So here at UC Davis, we have the Center for Spaceflight Research. And we are integrating the frontiers of a broad range of disciplines because it takes them all. Everything is going on at UC Davis. It takes to do spaceflight safely and productively. Education, all disciplines of engineering, human physiology, neuroscience, behavioral psychology, human-machine collaboration, and so on, to answer the kind of questions I've just been asking and so many more, but more importantly, ask the future questions that will both enable the human exploration of hazardous environments and bring back to all of us the life enhancements that we really need. So we are the first generation of humans that could both ask and answer questions like this. So I hope you'll join me in this fascinating journey. Thank you.
Wow, wow, wow. You know, this was really impressive. I hope all of you enjoyed all the presentation. And as I said in the very beginning, it's not the $1 billion only, it's the quality of research and the impact that we at UC Davis make. And you know, this is just an example of some of the activities, and we have a lot more researchers working in many exciting areas. And I hope all of you are inspired to take it to the next level going forward. Office of Research will be always there to help you in all your efforts. The best way to get started to reach us is research.ucdavis.edu. We have all the information over there, and I would encourage you to partner with us in helping you propel your research forward. So with that, we will end the program. But before, I want to end the program by thanking all the staff from Office of Research who have worked behind the scene to make this program of this quality. Thanks, everyone.